It's indeed a great pleasure to see your enthusiasm and the curiosity to learn about advanced techniques in bioinformatics. And I assure you that these 2 hours which you have given to us will be really productive and informative to us. So, with this warm welcome, I would like to give you my introduction in brief. I'm Sharvari and I'm Technical Associate at BDG Lifesciences. And I have 12 years of experience in the field of biotechnology. I've been teaching Biotech Engineering College for 12 years, and now I'm associated with BDG Lifesciences. We conduct various workshops, trainings, and I would like to give you brief about the company's introduction. Then we will be proceeding for the actual topic. So I'll start sharing my screen. Yes, so let us begin with what is BDG Life Sciences. This is our company. We started in 2010, which is with its operations in the field of biotechnology, bioinformatics, and pharmaceutical companies. And our wide array of services include research projects, workshops, training online on site, workshops online on site, webinars, internships, contract research, seminars, and conferences. These are some highlights of the services which we offer on bioinformatics, basics and advanced, on structural bioinformatics, on CRISPR, MD simulations, genomics, proteomics, immunoinformatics, R analysis, data sciences. These are just the highlights of the topics which we cover. I would request you to go through our website. Links will be provided in the chat box, and on which you will get all the details of services that we do offer. We will be indeed in a great pleasure to help all the fraternity of biotechnology with our services which can benefit you. Yeah, so now I will start with the actual topics that we are going to cover. I hope I'm clearly audible. If there is any problem, please team let me know. Yeah. So I'll start with the topics which we are going to cover in this 2 hours. We will be talking about Sigma. That is very important and useful and fantastic tool for mutation analysis. Then we'll be talking about UCSC Genome Browser. That is an excellent map viewer tool. First, it was NCBI map viewer which was used, but now they have developed it to UCSC Genome Browser, and it is far more advanced. On a single entry, you get a lot of information about the map of the gene of the protein. And most interesting part of it will be, I will be covering coronavirus 2 with the help of UCSC Genome Browser. Then we will be dealing with primer designing. Then we will learn what is phylogenetic. Primer designing we will be discussing with primer blast. Then we'll be discussing phylogeny, and we will discuss with clusteral omega as well as mega, which is newly introduced tool and which is also very efficient. Then we are going to discuss proteome analysis, that is protein-protein interaction with the help of string. And then we will predict the function of the protein with the help of, again, very efficient tool or consortium, which is known as InterProScan. So let us proceed with what is AlloSigma. AlloSigma is basically a server, or it is a tool for estimating the allosteric free energies acting on a single residue as a result of either ligand binding or mutations or both combined. Sorry, getting me. First of all, try to understand meaning of allosteric. Allosteric is when you have ligand and receptor which are bound to each other. But apart from this, if there is some other site and to which this binding and to which this residue is binding or mutations are occurring, and because of this binding or this interaction, there is effect on the you can say allosteric free energies on the actual site. So that is the meaning of allostericity. That means it is nothing but estimation of allosteric free energy acting on a single residue as a result of either ligand binding, mutations, or both combined. These allosteric free energies, they quantify the strength and the sign of the allosteric communication that is associated with effector binding and or mutations. Now, what do you mean by statement? Let me make it simple for understanding. Allosteric free energies quantify the strength and the sign of allosteric communication. That means the free energy which is associated with this allosteric reaction is estimated, and the is sign will depict the strength of allostericity. So if it is positive, then interpretation is different, and if it is negative, of course, when we talk about free energies in bioinformatics, you all must be knowing that negative sign always depicts the greater sign, and positive sign always is less preferable or favorable energy, right? So here, basically, we are going to compare the dynamics in the unbound, that means unperturbed. What do I mean by unperturbed? Undisturbed. So here we are going to compare the dynamics in the unbound or unperturbed condition and the bound or perturbed condition. And then after doing this, per residue allosteric potential will be evaluated. This potential indirectly, it is giving us the elastic energy, which is associated with the local structural changes induced by the protein dynamics, right? So, per residue, after unbound and bound, we are going to consider two different conditions, unbound and bound. And for each residue, we are going to calculate allosteric potential energies by averaging over all possible neighbor configurations of the residues in bound and unbound. Average of all these will state the per residue allosteric free energy which is involved. Okay, and with this what are we doing indirectly? We are indirectly measuring the work exerted on a residue of interest as a result of allosteric signaling produced by effector binding. So is the concept clear? What are we doing here? How are we analyzing the mutation? Is by giving two different conditions, perturbed and unperturbed. And in both the conditions, we are calculating allosteric energy which is involved. And calculation of this allosteric energy which is involved due to these changes will give us how these allosteric binding of or the mutation is affecting the dynamics. Okay, yeah, so pictorially or diagrammatically it can be seen like this. It is ligand free system, this is ligand bound system, and this is the allosteric potential. So as you all can see here, these are the residues which are involved, I, J and K. So different symbols, what they are doing is they are depicting the changes in the displacement. Okay, so you can see here I, J, K in circle are the reference atoms. Then I, J, K in width of the blue symbols are displacement change from I to I. You can see here from I to I in blue color is the bound system change pairwise distance. Clear? J, this J in pink color is free system change pairwise distance from J to J. Okay, so this is the displacement change, and K to K is again displacement change with the bound system. So this is nothing but just a pictorial representation of what I was trying to say. From bound to unbound, displacement changes with the reference system are depicted here. So, as I explained you about what is the concept of allo sigma, you can think about its applications. So, its application will be for the researchers mainly who are interested in fast estimation of allosteric effects. So, whenever you are studying any change, it is always advisable to see its allosteric effects also. So, this is very fast and efficient tool, I would say, to study binding or effector binding and mutations. So also this is important when you are studying binding energy associated with the residues. Also, researchers who are interested in mutations turning residues into bulkier ones, there are two types of systems which are also very interesting part of this tool is up mutation and down mutation. So up mutation will be for the researchers who are interested in mutations, turning the residues into bulkier ones. There we are going to, I'll show you when I'm explaining the tool. Um, when you are interested to allow or study the mutations or replacement of residues by the bulkier ones, then we will use the up mutation framework. On the contrary, if one is interested to give the mutations or replacement with the residues such as alanine, glycine, then down mutation. Okay, so now I have taken one case study that will be used in order to understand all the applications of the tool. So we will take an example of tetrameric enzyme phosphofructokinase. Okay, so what is the mechanism of phosphofructokinase? This enzyme is allosterically inhibited by phosphoenol pyruvate, and it is activated by by two ADP bindings to two overlapping binding sites. It is cooperatively regulated with respect to the binding of two substrates, fructose 6-phosphate in the presence of PEP. So this is the actual mechanism of PFK. Now we are going to study it with the help of allo sigma, which changes are which changes are giving you the results or changes in the dynamics, okay, allostericity. So again, I'm repeating, phosphoenol pyruvate is all actual in scenario. It is inhibited by PEP and then it is activated by two ADP binding to two overlapping binding sites. It is same time, it is cooperatively regulated with the respect of two binding to two substrates, fructose 6-phosphate in the presence of PEP. Okay, PDB ID is 3PFK. Yes, so what I was saying pictorially, this is our wild type residue. Then this is up mutated regulation of residue, this scattered lines, and this is down regulated or down mutated. This is wild system, this is mutated system. Now, what is the input data for the tool? It is very simple. This is very user friendly. Only the thing is you should be able to properly read the result and interpret the result. Otherwise, tool is very simple to operate. So this is input data. At the first step, user is asked to provide a PDB ID for the protein of interest. Alternatively, you can have a choice to upload a protein structure file in PDB format. Yes. So when you enter Allo Sigma, this is the home page of Allo Sigma. As I told you, here you can enter your PDB ID. So since we are using, as I told you, phosphofructokinase, you can enter your PDB ID over here. Then you simply submit the PDB query. After clicking submit PDB query, we are going to come across this thing. So biological assemblies and homologs. So first the server, what steps it is going to do, that I will tell you first, then I'll show you how it will look in the actual scenario. So this all today we are going to cover all the tools of advanced bioinformatics, but in 2 hours it is only possible to give you like theory and how does it work. In actual workshop we will be doing it hands-on, so you will be performing all this, you will be getting your results with the help of exercise, and so that is going to be hands-on. Today I will just make you through the journey of all the softwares which are used in advanced technologies. Okay. So after the sessions, if you join our workshop, you'll be more than glad. After each session, we are also going to provide you with the recordings as well as the notes for each tool. So it will be really productive and informative for you. Yeah, so coming back to this biological assemblies and homologs. First step is the server connects to EBI PISA database. And from this PISA database, it uses the best matches or assemblies, you can say. You can call them as assemblies. This will give you the best matches that correspond to the PDB entry which you have given as a query. So, after comparison of this PDB entry, which is given by the user, complete an ordered list of PISA database. Complete entry of the PISA database will be given, and we are going to call them as assemblies. This is the entry according to the, what you can say, solvation energy. Solvation energy means, in short, energy which is required to solvate the protein. It is nothing but your free energy. So with the according to the values of solvation energy, favorable is the value, the assembly will be at the top. So like that, we'll get list of assemblies, and then at having after obtaining this energy, you can, or a user can select the assembly of interest. Okay. Now, after selecting the assembly of interest, what you can do is, by clicking, you'll get the header of each column in the list. Okay. You will get the header of each column in the list, and explanation of the corresponding parameters will appear in the pop-up window. So here we will see this kind of result you get after entering the PDB ID as 3PFK. So this is your job ID. This is the structure source. These are the possible homologs. Can you see here? Yeah. So now, after processing is done, you will get these parameters: source, MM size, energy, formula, composition. So these are all PISA database entries, and this is what is important. This is according to the solvation energy, minus 97.21, minus 35, minus 31. Okay. Now, when you have this, you have three options of study. Okay. So first is binding sites and mutations, that computes the free energy response due to ligand binding and point mutations. Second is allosteric signaling map, which is allows exhaustively computing allosteric modulations of each residue due to single mutations. And then allosteric probing map. This is going to give us computation of allosteric changes due to the small probe. That means we are going to deliberately involve or deliberately give the sequence of some probe instead of mutation, instead of single residue, or instead of binding site. And we are going to study the changes or allosteric changes because of binding of this probe. Okay, so as I told you, when you get this assemblies, you will select any particular assembly. You will click on any particular assembly with the circle box, circle over here. After selecting assembly, you can choose between these three modes as I explained you. Depending upon what is the purpose of your study, you can select any one mode of operation out of these three. So first we'll go with binding sites and mutations. What does this, what is the purpose of doing this or what it does? Binding sites and mutations, they perform a single computation of the energy response to specified ligand binding mutations and or their combinations. Allosteric signaling map, it computes the free energy response to the mutation of single residues along selected protein chains. Okay, and allosteric probing map, it computes the free energy response to the binding of small probes along the selected protein chains. So first we'll see binding sites and mutations. It will allow the user to perform the single computation of the energy response to each residue after ligand binding or mutations. Now, once the assembly is chosen by the user, a panel with the protein chains and protein residues is provided on the left side of the page, like this. Okay, wait. Once the protein assembly is chosen by the user, a panel with the protein chains and protein residues is provided on this left-hand side of the page. And here, one possibility or one facility is there. You can see this is the panel which comes after you select the assembly. As I told you, we select with the circle first the assembly. After that, you will get this. Now, out of this, if you are interested, you're not interested to study whole protein, you can select only one chain. You can deselect any one chain. So here, what I mean to show you is all the applications, how to use the tool for the betterment of the analysis of the data. We can deselect any chain from the calculation. How to deselect it? Just click on the square box of the particular chain. Okay, now in this case chains can be directly excluded, and here you can see there are some discontinuities in the sequences, and those discontinuities, they are nothing but the missing fragments or residues. Okay, now there is this tool is very efficient, but it has the memory constraints, so server uses only the structures with up to 5,000 protein residues. So this is what I explained. You can deselect the chain. Now, after that, you get the list of retrieve binding sites in the right panel. These sites can be considered in the calculation by clicking on the corresponding buttons. Okay. So after the selection, it will turn red like this. So you have this chain A, this chain J. These are the list of binding sites which are retrieved from the assemblies. Now, if you select particular binding sites from this list of binding sites in protein homologs, whichever site you are selecting here will be turned red. Also, the residues which are involved in this binding sites will be turned red in this chain. Okay, so this is one more thing that you have to do. First, we selected the assembly. Then we are going to select the binding sites. So here the figure is showing, as you all can see, 4 binding sites PGA are given here in this diagram. Now the calculation is done for this particular selection. Can you see here? These options are there, and here on the on the right top corner, you have this run analysis button. You can click on that to start the computation, right? Now it will start computing the effects on these binding sites. Understanding? One more facility is there. As we have selected here the retrieve list, sometimes the researchers can be interested to use their own binding site. This facility is also provided by the tool, and that is known as user designated binding sites. Besides the available binding sites which are obtained from homologs, user can manually define the binding site according to the procedure. So here you can see you can just select the residues over here in the select residues. Okay, if it is user designated binding site, you can select the residues manually in the left hand side of the panel. And also you have to type the name of the site here in the site mutation name. You have to type the site mutation name and click the site button. Click the site button over here and it will turn red. After clicking on the site, it will turn red, and then you can select or you can click on the run analysis button. So you can provide your email address also, so the results will come by notification to your mail address. So you don't have to really wait, or this is a quick tool, no problem about the timing, unlike some tools which are there in computational biology like FIRE2, which takes a time of 5-6 hours. There you can't wait. You have to submit your query and get the notification mail of results. So here you can have that facility. After running analysis, you can get the results by mail. So understood. First was just the list of just using the list, and the second is to use the user designated binding sites and run the analysis like this. So as I told you, you can select the residues over here, and you have to, in this, you have to click the site and site mutation name. You have to enter the site that will turn red and it will say site 1. Then you have to click on run analysis. Same way, you can do user designated mutations. So mutations can be also done in a similar way. So as I told you, very interesting part of this is up mutation and down mutation. So for this, what you have to do is you have to select the residues same way like this. You have to select the residues in the left panel, provide the name for the mutation over here, make it a mutation. So you have to change the residues over here. To click on the residue which you want to select, and you have to type the selected residues in this box. Then you have to give the site mutation name, make it mutation and start computation. But before that, you have to mention or you have to click on either up mutation for up mutations or down mutations for down mutations. This will add a red button with the name of mutation set, and corresponding residues will be turned red in the left sequence panel. So best part of this tool is also wherever you're doing any changes, it will be highlighted by color change from blue to red. So there are less chances to make any mistakes by like you can say ignorance. So up mutation and down mutation. Then again, you click on the run analysis button. The same thing which we discussed, I put it here, that we are using for these 4 specifically binding sites. Then the computation result. Okay, so we are done till here. Now you understood what are the applications and how to use it. Now what you get as a result. So let me explain what are we going to get exactly in the result. Structure view panel. This is the structure view panel. Very interesting, right? It is in the form of tube, and it is this type of depiction is known as PV color tube. So there are colors which have different significance with respect to delta G. See here, pointing the, here you get the effect of mutation, and the right panel and left panel. You have right panel and left panel. Effect of binding mutations are shown according to the color codes. Here, strong conformational changes or low are used. here, you see here, you can hover over to highlight the binding site residues on structure. So gray color will give you, wherever you're hovering, that time gray color will give you those particular binding sites. Okay, and then we can click to show the residues of the sequence over here. And this blue and red are depicting higher and lower energies. Also, you can click on high delta G and low delta G, which if you click on high delta G, it will give you the residues which are showing high delta G in this chain A. If you click on low delta G and ask to show the effects on the site, it will show you the residues with high delta G and low delta G in the result. So basically, after doing or after giving input of the data, when you get the result of the analysis, we get it in the two types, structure view, and this is the effect. So structure view, you get this tube-like structure where wherever you see red color, it is to the low delta G, and wherever you see blue color, it is to the high delta G. So also, to get more specifically, you can click on high delta G and then you can click on show effects on the site. It will be depicted over here in the structure view as well as here in the chain view, chain A and chain G. Clear? So this is how you can read your result. And this is the modulation graph. From this data, modulation graph is also obtained, which gives you this kind of graph. So residues and its delta G. You can compare it for 4 different binding sites. Clear? Yes. So I think this tool is clear to you now. You can use it for your purpose and explore it. Yeah, so the next tool that is also very important and vast tool, you can say, it has vast applications, which is UCSC Genome Browser. UCSC Genome Browser is you can say it is a dynamic resource that allows user to perform the curiosity of about studying any particular protein at gene level or genome level. Also, it will give to us the facility, or most important application of it is to test the hypothesis of unknown protein. So it has got a lot of application, and we are going to focus on use of UCSC Genome Browser for coronavirus. Okay, so once you enter this UCSC, that is University of California, Santa Cruz Institute, UCSC Genome Browser page, here you have these entries on the web page, on the first page. So this is the display feature here. Because of the development of huge amount of data in coronavirus, UCSC Genome Institute, they say that that data generation inspired them to make a specific page on study of coronavirus data. So as we move along with the slides, you will understand how efficiently and what amount of efforts must have been taken by scientists in order to compile the data of coronavirus in all the species. So that is indeed a very great help to a research which has been carrying out in the coronavirus field, right? So if you click on this particular coronavirus data, it takes you to this page: COVID-19 portal, browser paradigm, viral assembly names, SARS-CoV-2 datasets, and UniProt tracks. We are going to see. Also, you have detailed pages of annotation, configuring track display parameters, NCBI genes track, RT-PRC primers, and we are going to also get the information about, this is newly introduced thing, which is excellent, all the vertebrates which are infecting coronaviruses. So let us start with one by one. COVID-19 portal first. So if you If you click on that, it will take you to this page, COVID-19 pandemic resources at UCSC. So it will give you basic information about from where it started and how it spread and which are the institutes which are involved in the research of the same. Then it will take you to this page. Here, if you click on the left panel, you can say here, then it will take you to the, after clicking this left panel, it will take you to this information of UCSC Genome Browser on SARS-CoV-2 Jan 2020, its accession number, assembly and move code. Okay, right? So this gives you 29903 base pair of gene or genome of the coronavirus 2, SARS-CoV-2, right? And this gives you, this page gives you information about all its, you can say, information about this RNA as well as information about all these protein products. In the black color, can you see? All these patches of black color are nothing but the protein products. So this is the page. You can see, you can hover over this. This you have to do on yourself. That means you have to spend really good amount of time and patience in order to study this one page. So if you come to this, you can hover over your mouse to all the entries. It will take you to the detailed information of each and every entry. Yeah. So now these entries in blue color are nothing but your gene, its location. You can see here this location is 5,000, 10,000, 15,000. So along with its particular location in the form or in the, you can say in the order of base pairs, it is also giving you the information about its location. What does it mean? Here also, all the information about signal, peptide, transmembrane domains, disulfide bonds, protein domains, every information along with the its location on the, that is why it is known as genome browser or map viewer also. That means the particular location on the genome. Here the protein products are also given. So like that you can study and interpret your data. Now the browser paradigm or basic paradigm. It is nothing but a set of coordinates which represents the viral sequences and important sites or biologically significant sites. Okay. So this is the basic paradigm. Again, as I told you, you can also do one more thing. If this is a whole genome, you can select a particular genome by just clicking and dragging the particular region. If you click on this and drag a particular region, you have the facility to zoom in and zoom out. And that particular region you can study. Okay. Yeah. Then the next part is you have, as I told you, it is a dynamic resource which gives you information about all possible biologically significant information. So this has, you can see these boxes which are based on the applications. Once you scroll down over here on below this page, you have all these possible applications: mapping and sequencing, genes and gene predictions, UniProt, immunology. You can imagine how vast, and at a single go you're getting so much information. So for example, I'm selecting here UniProt annotation. So it will give you all the UniProt entries and all the information about it. You have facility to see RT primers. So if you click here on primers, it will give you information about the primer designing also. Only the thing is you should know how to operate. So click on the particular application. You have to click on this. You can see here hide option in there. Instead of hide, you will see in the drop down pack option. So you have to click on that pack option. It will take you to the corresponding information. I'll see if if time permits, I'll give you live demo of UCSC, because we have to cover lot many tools. But still, if we have time, we can cover live demo of 1-2 tool. And also I've repeatedly been saying that if you join this workshop, we have 10 days workshop, you will get actual hands-on experience of doing it. Also you'll get the notes and video sessions or recorded sessions of all this. Then you go for viral assembly names. As I told you, you click on viral assembly name, nothing, it is just giving you this name of the assembly at top, WCOT. So it started from WCOT. SARS-CoV-2 datasets. As I told you, if you enter or if you click on here, you can get all the data sets of SARS-CoV-2, all the proteins. It's what you can say, spike protein, then S-glycoprotein, then membrane protein, all the information about its location. Here, nothing. The color depiction I have talked about. Here, the blue color gives you information about the signal peptides. Primers, as I told you here in this page, whatever application you need to check, you can click on there. That if you want to design a primer, you go to that primer section and instead of hide, you select pack. So you get this RT primers. Yes. Now the last important application, which is vertebrate infecting coronaviruses. If you are interested to know which all organisms can be infected or which have shown the history of infection of coronavirus, okay, those are vertebrate infecting coronaviruses. So these are the vertebrate infecting coronaviruses. You can see here, it may appear little scary in the beginning to after seeing, but just the thing is you have to understand how to analyze the result. Here you can see all these colors, right? So here on the left hand side, you have description, like which entry is that. So here you have all the species, like Turkey COVID. In the Turkey, which type of strain was developed in which year? 2012 strain, Germany 2012. Okay. So like that, all the entries of human COVID HKU1. So that means all the similar strains of COVID-2 are aligned. This is nothing but multiple alignment of 119 strains. For getting me? So 119 strains are actually given in the form of result to understand which are the viruses or outbreaks which are similar to coronavirus 2, which has come in 2020. So you can see here 2015 one virus was similar, 2012 one virus was similar, in which species, that also is given. You can see here this green color symbols are non-significant replacements and this red color are significant. Also you can see here these spaces. These spaces means there are missing residues when you compare these two types of strains. Okay. So this is about the information about the vertebrates. Now if you click on this single entry, it will take you to the information about that particular outbreak and that particular virus. Here, like this. Fine. So isn't it a fantastic tool? Yeah. Okay. So here we complete UCSC genome browser for coronavirus. As I told you, it's just a trailer. There are so many applications in this page itself. If you decide to get information about each and everything, it is going to give you a lot of vast information about the data set. You can try it for yourself. Then I'm going to come to one more important application of this, what you can say, computational tool, and that is primer designing. Okay, so primer is basically a short stretch of sequence from where your actual sequence starts. Okay, so it is very important, you know, in PCR also to design a primer. It is got a lot of importance. So how do you design primer? By using NCBI, that we are going to see today. So I think most of you are from this biology background and you will be aware of this basic database of NCBI. NCBI is National Center for Biotechnology Information. Here you have, it is like a consortium and it has vast application as it is a compilation or consortium of sub databases, many databases, gene, protein, genome. So here how to operate the NCBI is the basic part. You can have in the drop down whichever database you want to go for. So here I've taken example of human albumin. So I will select the gene in this and I'll simply type here albumin homo. Okay. Then when you click on albumin homo, we will come here. So its location is given, which is 4q13.3. 4q13.3 is its exact location and its binding pattern. Okay. Then you have exon count. How many exons are there? That is also given, and that is 15. Okay, so here you have 15 exons for this particular human albumin. Then the next click, you will be taken to this 15 exons which I was talking about. If you count here, 1, 2, 3, 4, these are 15 exons on the genome. As we discussed in UCSC genome browser also, this kind of depiction is nothing but your location of each data, each base pair. If it is RNA, it will be a single base. If it is DNA, it will be base pair. So in the form of kilo base pairs, this sequence is given. Okay. Now here you have these 15 exons. Now if you are interested to find a primer against a particular exon, you will click on that particular exon. For example, if I click on this exon, I will be taken to this entry. Okay. Next page where I have this exon. It is this exon which I have selected. If I click on that, I will be taken to this page. Here you have this exon. Then I want to design a primer against this exon. So what I can do is I will select a region which is behind and which is forward to this area. Okay. So for example, I've selected this exon. It is always a good practice to leave 40 to 45 bases behind and forward, and then select a particular region for primer designing. So you have to, what you have to do, you have to leave 40 to 45 databases and you have to click here and simply drag your mouse from in the forward direction over here, and you have to leave 40 to 45 bases here and simply drag your mouse in the reverse direction for reverse primer. Clear? After dragging your mouse, you will get on the right click, you will get the option of blast and primer search. Clear? So after dragging or selecting a particular exon for which you want to design the primer, you will click on blast and primer search. After clicking on blast and primer search, you will be taken to this page where if you want to give certain, what you can say, certain parameters, you can change it, but by default, we keep it as it is. And then you have to click on get primers. Once you click on get primers, you will be taken to this page. So it is a graphical view of primer pairs. Fantastic. You on a single go, you have to just carefully, you have to select the area of exon which you are interested to find the primer. After that, you will get this list of primers. Primer 1, primer 2. So primer 1 pair. from here to here with the location on the chromosome. So this is primer 1, primer 2, primer 3. Usually we select first primer. So primer 1, if it is selected, it gives you detailed primer report. So this will tell you what is the sequence of the primer in forward direction, what is the sequence of the primer in reverse direction. Fantastic. Primer 1, 2, 3, like that, you will get the detailed primer report. Accordingly, you can design the primer. So here I had taken one example, and this is also the option. In the same home page, you have to give your entry. Here I have taken example of IL-6. You can put your entry as Homo sapiens interleukin 6. You have to just give a right click. It will take you to these options in the red color. Then you have to click on pick primers. That is also going to give you the same result. Only this thing will be like, it will be depicted like this. Okay, fantastic. Yeah, so we learned 3 important tools which are easy to operate but gives you a lot of information. First, we have seen AlloSigma. Then we saw this UCSC genome browser. Then we have seen this primer designing. Now let us move on to the next concept, which is very, very important. Phylogenetic tree. What is phylogeny according to you? Phylogeny is basically, it is the depiction of order of evolution, isn't it? So how to understand the evolutionary order by means of relatedness of the sequences of proteins or nucleotides is nothing but your phylogram. Why it is named as phylogram? Because it is in the form of tree. Some important terminologies I'm going to tell you in short. What are the branches? Branches are the splittings from the common root. Root is the common ancestor. From the root, the branches are splitted, which will be telling us, if we put our species on these branches, branch length is going to tell us, or the equivalence of the branches in 2 species is going to tell us what kind of relation they show. Do they belong to same clade? Clade means same group. If they do not belong to same clade, how much distance they are covering from common root, all that we'll get to know. And then node. Node is a point of splitting. At which point these species are splitting, that is called as node. So this is a simple depiction of tree of life. These are the branches of the tree. So very closely related species will be there on the same node. This is node. And distantly related will be on the large distance. So you can see here humans and chimpanzees, they lie very close to one another, right? And here you can see jellyfish and human, they are far apart. So they are lying on far branches. As you can see, this tree or this complicated branches, they evolve from the single organism, single cell organism. Okay. Yeah. Now how to use this? We are going to learn 2 tools, Clustal and Mega. Clustal has been used from many years. Okay. When we developed this pairwise sequence alignment, multiple sequence alignment, pairwise we use BLAST, multiple we use Clustal. Now from the date of Clustal, phylogram is produced, and that tool is known as Clustal Omega. Now here in the window of Clustal Omega, you put all the species, FASTA sequences of all the species. FASTA sequences meaning I believe you all know, FASTA sequence is the sequence which is always starting from, and it is a standard symbol which is used by all these tools or softwares in bioinformatics. So it starts with greater than symbol, then the string of letters. You are not supposed to make any mistake when you're copying and pasting the FASTA sequence. So here in the input sequence, you have to enter how many species that you want to study. So here I have taken one example of gamma fibrinogen. in 4 different species. So I have pasted at the time, I have downloaded FASTA sequences, and then I've pasted all the sequences over here in this window. After pasting the sequences, you can also upload a file. You can prepare Word file and then you can upload file. Not a problem. After that, you have to just click on the, sorry, you have to here at the down, you have option to submit. You have to simply give a submit. After submitting, after some time, you will get this alignments, result summary, guide tree. Today's focus is to give you understanding of phylogram. So I'm not going to go into all these details. Alignments you can see in multiple sequence alignment, then result summary, then guide tree, phylogenetic tree, etc. So we will be clicking on guide tree or phylogenetic tree, and we will get this tree. Now, how to read this tree? That is the important part. So here you can see, for simplicity and your understanding, I have just taken only the 4 species. You can do it for 10, 15 species, okay? So I've taken only the 4 species, and then I've clicked on guide tree. So I get this kind of tree, which is also known as cladogram. Can you think why it is named as cladogram? Yeah, because in the previous slide, I mentioned about clades. So clades are nothing but the group of species, which a group of the single group of 2 species, 2 or more species, which belong to common ancestor and which share high amount of similarity. So the tree which is made up of this clades is nothing but cladogram. So it is nothing but a similar synonym of your phylogram. So then you get this cladogram. These are the names of the species. Now these are the accession numbers to UniProt or SwissProt. You can go back. These are some common names. Actually, I have learned it by name because I use this tool very often in the workshops. You can study this accession number and go back and check which is the common name. So this 2676 is the coyote. This is tapir, pecta, and CMDR. So chimpanzee and peccary. These are 4 species. Now, how to read this is, if for simplicity, I name them as 1, 2, 3, 4 for now. Okay, so 1, 2, 3, and 4. So you can see here, these 2, 3, and 4, they are showing equal branch length. So they are belonging to one clade. So, and also the branch length is same. So they are belonging to one clade with the equal branch length and at a same distance from this node. These points are nodes. This is the root from where tree is splitting. These points are nodes. Okay. So from the node, they are splitting. So this 1, 2, 3, 4, 3 and 4 are, since they belong to common node and also they share equal branch length, they belong to common ancestor. Also, they have highest similarity. To this clade, second species is also related, but at a larger distance on a single clade. Second species and first species, they do not form clade with any other species. So they are on simple or they are on different branch. Understood? So this is how you read the tree. How will you interpret the result now? 3 and 4 are highly similar, belonging to common node with the equal branch length. Second and first are similar to third and fourth and also to each other, but at a larger distance since they belong to different branches. Always remember, when you read the tree from horizontally, it is the time scale and order or rate of you can say time of evolution, and vertically it is always the relatedness or similarity. It is the percentage identity or percentage similarity. So this is just a phylogram without branch length. There is option of getting phylogram with the branch length also. Okay, so the branch length option is nothing but here these branches are showing only the similarity or relatedness, but when it is according to the branch length corresponds to the time of evolution, that time this particular horizontal branches are given according to the scale length. So those scales are selected and actually scientists put the scale, compute the value of evolution, and the branch length will correspond to that particular value of the time of evolution. Now, this is the next tool that I want to give you information about, Mega X. So now the cluster list, we simply have to go to Google and put cluster omega, okay? And that will take us to this page which I've shown in the beginning, okay? Then this is online, free online tool. Mega is the software which you have to download. So you'll go to Google and download Mega X. Before that, you will go to NCBR. Here I've taken one example of K-lactis 26S ribosomal rRNA gene. You can take any other example also. So if you install or download Mega X, and like other softwares which are locally installed, you have to just click on Mega X, go on next, finish the wizard. It will be locally installed on your computer. You have to just open it, run as administrator. It will start operating. So this is the home page of Mega. Here you have these many options. Now you have to go to file over here, and once you click on the file, it will ask you to download the FASTA files. So before going for operations here, you have to make one file. You can name it as mega sequences, and you have to give some 4-5 sequences for alignment. One by one, first you will select one file, open it up. It will be opened like this, like this. Then one by one, you can select all other files from any folder. You have to go to edit option over it. Can you see edit? And this edit will give you option of insert sequences from file. You have to select a particular file and insert all the sequences. They will come like this. After that, you have option of phylogeny. You have also these many options. You have option of align also. You can make multiple sequence alignment like we did for cluster. And you can also use cluster from Mega also. Here in the alignment, you have two possibilities of aligning by muscle and by cluster. You can select any one and take the multiple sequence alignment. Then the next option is phylogeny. You construct the phylogeny. Click on this cluster phylogeny construction. You will get this phylogeny. Fantastic. So this you can compare with the cluster. It is the same type, same way you have to read it. Here, this is the thing which I talked in the before. These are the scale values. Here, Mega gives you branch length, which is scaled. That means this particular distance shows here the distance of a particular clade from the node. Equal the distance or equal the branch length, highest is the similarity. Fine. So this was one more important tool that we discussed. It's the phylogram. Very, very important from point of view to study the functional similarity, to predict the structural similarity, to explore which proteins are similar to your protein, and then you can shortlist them for further studies. Very important application. Then next, we are going to discuss string. String is basically for protein-protein interactions. So whenever you are talking about proteome analysis, protein-protein interaction is indeed a very important part. To study protein-protein interaction, this is a fantastic tool which is known as string. Now let us see how to operate string. This is the home page of string. You have to simply type string in Google. It will take you to the string. This is the home page. This gives you information about how many interactions are there, how many organisms are there, how many proteins are there. Yeah. So this is the home page of string. These are the data sources. Genomic content context predictions are there. High throughput lab experiments. These are the 5 sources from where this study of interaction are done and put it in the form of database. So what are the sources that they have mentioned on the home page? Genomic context predictions, high throughput lab experiments, co-expression or conserved expression, automated text mining, and previous knowledge in databases. String database, currently it has around, you can say, this around very high number of proteins. It's 9643763 proteins from 2031 organisms. Now you go to string homepage where you will enter the protein name, as I have taken this example SLC6A1. Okay, SLC6A1. Once I put here and go for search, it will take me to this page. So it will give me all the entries of all the species and its related protein, or not a related protein here. It's protein which is showing interaction to your query protein. Okay, so these are the other species or organisms along with other proteins which show interaction with the query protein. So you can click on specifically Homo sapiens. That will give you all the proteins which show interaction to your human albumin. Then you can select mus musculus, you can select any other species. But when you select the protein species or protein from the checkbox, it will give you only the limited result for that particular entry. Yeah, so this gives you the network or the analysis of results. SL6A1 is our query protein. These are the other proteins which are showing interactions. This is known as first shell. That means first shell of interactions. Topmost proteins, 10 proteins which are involved in the interaction with query protein. Okay, so most important 10 proteins which are involved in the interaction are mentioned in this diagram. You can see this ball kind of structures. This red ball is our query protein. Other proteins are the interacting proteins. So all these are interacting proteins. Fine. So this red color ball is our query protein. Now we can drag this and see it like this. Yeah, I have dragged this protein of the query, and this is the first shell. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All the proteins like GABR3. This is the protein which is in GABA receptor protein. All this information is given in this. Predicted functional partners. They are called as functional partners. So all the, here you can see GATs. Its information is given. And these numbers are nothing but confidence levels. Confidence levels have to be higher than 0.90 so that it's always best to select the proteins which are interacting and showing confidence level more than 0.85. So all this confidence level, they depict the proteins. You can select or click on each of this entry and it will take you in which pathway it is involved to your query protein. Fine. Now these filled balls are always depicted when information about that protein. If you click on this filled protein, it will take you to its 3D structure as well as its information. These empty balls are the proteins which are not giving us any information about its 3D structure. So these are the colors. Also, they are given the color codes to interactions predicted with higher confidence level, with lower confidence level, like that. Okay. Now, as I told you, If you click on any particular protein or the emptied ball, which has all the information associated with it, if you click on it, it will take you to this page. It gives you homology model of that protein, 3D structure of that protein. Then there are these options like network, experiments, database, text mining, neighborhood, co-occurrence, co-expression, fusion. You can click on each of this. Each of this has different applications. So network, if you click on the network, it shows you the current interactions, which is shown over here in this diagram. Nodes can be moved, pop-up provide information of each node and each edge. Experiments give you information about its source, like which experiments are performed, co-purification, co-crystallization. Databases gives you information about the protein in other databases. Then you have neighborhood, like what are the groups of genes which are similar to the your to your query gene. Then co-occurrences, which are the genes which show co-expression. Co-expression and co-occurrence are similar, more or less similar terminologies. Their co-occurrence basically gives you names of the gene families which are, you can say, occurrence which show co-occurrence pattern which are similar to the genes. Co-expression is which are observed to show expression with each other. Okay, so like this I had taken the graph of co-expression. You can see here I put this horizontally and put this vertically. So you can see SLC6A1 is co-expressed with SL6A1 with the red confidence level. Then it is co-expressed with GABRF5 with the light red color. This is with bright red color. So like that, this observed co-occurrence can be studied. Okay. Yeah. So this was about the string operations. How do they interact with one another? This is important depiction of string. You can put your query, and here, since it is a results screenshot, I can't move along with this. When you are performing it, you can drag this and you can study all this network of association of interactions. Yeah. Then we will go ahead with the next tool, which is InterProScan. Yeah. So the main function of InterProScan is the function prediction. Okay. We are going to predict function by using InterProScan. Again, InterProScan, you must have observed all these tools which we have talked so far, and also other tools which comes in basics of bioinformatics. They are not really very difficult to operate. When does the difficulty come is not exactly, I will not call it as difficulty also, little bit of expertise and, or you can say, tricks to handle that comes in operations like docking, QSAR, simulations. But all these tools which are there for basic study, primary, secondary structure, function prediction, genome analysis, homology modeling, all these techniques, they are user friendly and almost all are online except for visualization tools also like Swiss PDB Viewer and Rasmus. They have to be locally installed. So all other tools are same to operate. You can say they are simple to operate. Now this is InterProScan and it is for function prediction. InterProScan is also excellent tool, and in the single go, it is redirected to many databases. That is the best part of InterProScan. So it is called as consortium. What do we mean by consortium? Consortium is like a compilation of many things together. So this is the simply you have to type in Google InterPro. It will take us to this InterPro, and here in the window, as I told you repeatedly, we have to enter the sequence in faster form. So here I have taken example here. You can see its accession number. It starts with greater than symbol. Its accession number, its entry, and its species name. After that, it starts with the actual sequence of the protein. Protein which I have taken here is gamma fibrinogen. So gamma fibrinogen sequence is copied and pasted here. Then here the other option is also there. You can choose a file which is saved as a Word file in the form of FASTA sequence. Then you can click on search. Once you click on search, it will take some time of 5 minutes to load the result. After that, this is your result page. Okay. Now let us learn how to read this result page. So you have this up topmost is your query sequence, and the bars which are down, you can see different color bars. Those are bars which will redirect you to the particular database. So, and at the down, you can see over here biological process, molecular function, and cellular component. So you can understand this is our gamma fibrinogen. Biological process it has given, it is involved since it is a fibrinogen. It is involved in platelet activation. Then with the GO, what is this GO? GO is the accession number for particular entry. That means it is such a superb, what you can say, tool that if you click on this platelet activation, even if a person is very new to the field, that person also will be able to study this database and get the information whatever he or she wants. So biological process, you simply click on platelet activation. If you click on platelet activation, it will take you right from the meaning of what is platelet. Platelet activation, how it is done, everything. So this is the particular accession number for that. GO, if you click on that, it will take you to the particular biological function, meaning of that function, and application of that function. Next is it is involved in. So in short, this is first giving you information about the protein. Your query protein is involved in which biological processes. So it is involved in platelet activation, it is involved in protein polymerization, and it is involved in blood coagulation. Then comes the next part, molecular function. Molecular function in which it is involved. It is signaling receptor binding. Then if you click on this GO number, it will take you to how it is involved in signaling receptor binding. Then cellular component. As it is a fibrinogen, it is showing us fibrinogen complex. If you click on GO, it will take you to the information about its function or its meaning of fibrinogen complex. Now the very important part comes here. Here, if you click on this first entry, it is IPRO entry. It's the query itself. It's interpro entry. Interpro entries are called as superfamilies and all others are called as subfamilies. Once this is the family. Once you click on this PTHR, PTHR is nothing but your Panther entry. PTHR is the Panther entry. That means it takes you to the Panther database. And what is Panther database? It is very popular and useful database. It gives you information about the gene families, subfamilies which are related to your protein. So if you click on here, it will take you to the Panther entry. There is option to go to Panther and it will take you to the information of Panther entry. Next comes here this PFO 870. This is nothing but PFAM. So it redirects you. If you click on this, it redirects you onto the PFAM database. PFAM database, again, it gives, apart from Panther, which talks about the gene relatedness, all other databases are mostly the protein function information or else the sequence information. So next is PFO. As I told you, it will take us to the PFAM database. its information in the PFAB database. After that comes SMO. SMO database is a smart database which gives information about the proteins entry in the smart database. Then comes this unintegrated part. What do I mean by unintegrated part? This is newly highlighted or newly entered feature in Interpro. Uh, Unintegrated means the entries of new proteins which are explored. So they have not yet classified in this particular databases. They are just like piled up in an integrated path, and scientists will then thereafter, depending upon its relatedness to the database, they will club it or they will compile it into the Panther, Smart, or PFAB, like that. Then the predictions, it gives you prediction, this black color region, and then this pink color region is also a little more of importance, is because this pink color portion gives information about disorder. That means sequence in the protein, which if you can say this shows disorder in the structures, which structures are important for us, like alpha, beta, gamma, secondary structure prediction. So this particular pink color is the region where if there is disorder in the structure, it gives disorder in the function. So it has effect on the protein's functionality or function. So this is the region which is responsible. You can say, if you just take it out, you can see 55 to 77. This region is important from structure and function point of view. Clear? Then I just want to give you some information about other tools also, some preliminary tools like Expacy and PepDraw, which are used for protein primary analysis. If time permits, I'll show you one example of that also. Now, since we have completed with the explanation part, I would like to give you demo of, let us take demo of any two tools. Hello Sigma will take a lot of time. So we can go for UCSC genome browser, and we can go for phylogram. So I'll stop here my presentation. I think theory part and the application part and the concept behind each tool must be clear to you. Now we'll see two demo. So the first thing that I'll show you is phylogram by cluster. As I told you, we will simply type crustal omega in the Google search. Here, as you saw in the presentation, we are going to enter any sequences. So for that, I'm going to use FASTA sequences. I already have file of cluster sequences in my computer, so I'll go for that. But when you are doing for the first time, you will have to download it from NCBI or Swissprot. Fine. So now what I'm going to do is I'll copy all the sequences at a time. Maybe I can take 4 sequences for simplicity again. You can take all. I have this 7 sequences saved with me. You can copy it. Paste it in the window of sequences. Here, as I told you, you have to set the parameters. You can just be for notification by mail. You can click on this. You can enter the email if you want. Click on this. This is what I mean by when we talked about alignments. So this is the alignment. Can you see here all these sequences? Now I will tell you how to read these alignments. Can you see in all these sequences R is conserved throughout, and this is the star symbol. Always remember in all these tools, be it a blast, be it a MSA or classical, star symbol always mean for the identity. Okay, so star symbol or asterisk symbols gives you identity. You can see here one dot. One dot is always a, or first let us understand two dots. Here you can see at some places there are two dots. Now see carefully. These two dots are L-I-I-I. So that means only in a single place L is present instead of I. I is conserved throughout and L is replaced in one sequence. So that means that particular place there is semi-conservation. Two dots always mean for semi-conservation. And one dot always mean for the slight conservation. So if you see here carefully, in this place there is one dot. So T, T and N. Two places there is T, two places there is N. So that is nothing but slight conservation. Understand? Understanding? This star is full identity. Then one dot is less identity or only the conservation. And two dots is semi-conservation. You can show us to show by colors, so it is easy to read. Understood? So this pink colors throughout, they are showing identity. Green color, identity. Different symbols, different colors are given. Now the part of our interest is phylogenetic tree or guide tree. Fantastic? Yeah. Now how to read this? Again, I will tell you with the help of this slide. This result. So as I told you, real will give you the scale values. And cladogram will give you without scale values. So see here, now if you remember and apply what I explained when we discussed it in theory, these two are the thing but same clade. They are highly similar because they show equal branch length. They belong to this node. They are coming from common node. Then comes the next species, which is on different branch, and then comes the next species, which is also on the different branch. So this is how you read the guide. Fantastic? Yeah. Then we can see one more tool, how to operate. We'll go for UCSC Genome Browser. You have to simply type in Google. All the tools which I've covered today, as I repeatedly said, you have to just go through the Google and get it. Okay, there was no tool which needs to be installed. Here, as I told you, coronavirus data. This is the information page. Then you go to Genome Browser. Yes. So get this genome. Whatever I explained, mapping and sequencing. Right? You can zoom out, zoom in, whatever applications I told you, you can do that. Okay. You can click on this left panel. All this, as I explained you, all this gives you, so this part, as I told you, if you put your mouse on the particular entry, replicase polyprotein 1A, then you can click on this. It will take you, see all the information which is given. Only you have to go to particular entry and give it, your mouse will be over in this rectangular box. All the information is given for that particular sequence. It is responsible for the cleavages located at the end terminus. So all that you can read. If you click on this, it will take you entry about the protein domains which are involved, uniprot entries, everything you get. And as I told you, uh, hide this, you can say full. Immunology, refresh. You have to add the package actually. Mapping and sequencing. Then here, as I told you, you can go for primers. Then you can go for pack instead of height. And then refresh. So it will give you information about all the primers. This is tedious to read, so take some time specifically for it and then go for it. Understood? So this is about operating UCSC Genome Browser. Yes, so now I think I have completed everything which was my target. Fine. So now I would like to know if there are any questions from your side, and I would like to talk about little some features and some important points. Uh, this all which we discussed is a part of advanced bioinformatics. Like this, we also have basic bioinformatics, and we have clubbed both things as basics and advanced bioinformatics of a 10 days. So this, please take benefit of this 10 days program, and links are there for upcoming workshops which our team is putting up it in the chat box. So all the schedule, fees, registration, how it is to be done, all the details you'll get on that. And please register soon, you will definitely get lot many tools to understand. And this was just a demo and theory and like a trailer part, whichever is possible to cover for me in 2 hours. I have tried my level best to cover it. But when you're doing it in the workshop per day session, uh, you will be doing hands-on. I will give you exercise at the end of each explanation, and after the explanation you will also get the recorded sessions or recorded videos of all the explanations, where you can always refer it for your when you're doing it on your own. All the steps will be covered, all the videos will be given, all the notes will be provided, extra papers along with all the resources will be provided, so you can understand how thoroughly you will understand each and every software and each and every topic. So this is about the advanced bioinformatics that we are launching in the January. You can get all the details on the chat box links that are provided. So please do enroll for it and take the maximum benefit of learning maximum tools in minimum time. Okay, so best part is you will learn right from operation of computer. I'll be teaching everything because some people are new and they don't know how to install the softwares. So we are going to, it will be totally interactive. You can go through the feedbacks and previous videos also. We have a trend of personal attention. I don't go ahead unless and until each participant has understood everything. So that is going to be really beneficial for you guys, so please take the benefit of that, and we will be glad to have maximum participants. I would say all participants which are attending today's session, if I would have them as participants in my workshop, it will be more than pleasure for me to teach them. So that's all from my side. I hope you liked the session, and if you I have generated what I can say curiosity to learn more about it. It's my success of the session. It's the success of the team which have taken efforts. And thanks. That's it. So if you have any query, you can put it in the chat box. So again, other workshops are also there on docking, simulation, everything. If you guys are interested in any topic, please refer to this website, and please do register for our workshops. Thanks a lot and have a good day and take care. Yes, team, shall I end the meeting then? If you don't have any question. Yeah, sure, we can wait for the questions. Since we have time, uh, yes, Priyanka, which part did you miss, dear? You want me to repeat? We have time. I can repeat anything for you. Okay, fine. You can check the video also as it is mentioned. But apart from the part which you have missed, if you have any query, then you can discuss with me in theory part or any other thing information if you want, you can ask me. We will wait for 5 minutes. Yeah, I think so. No questions.